Hey, congratulations to everybody. This is amazing. This was so exciting to see all these articles come out and to see the actual Nature paper I have right here. I don't know if you can see in my tiny video, but the real thing. It was very exciting. And we even got an email from Nature that they sent to Mala saying that they gave away hundreds of copies of the Nature cover of the Nature journal at SFN. And it was very exciting. And they even want Mala to like write a book now. So fancy. And one last thing, if you didn't see, Flywire even made it onto SNL, onto Saturday Night Live, So this fly's got jokes. Sorry. I try to say something funny, but I'm just winging it here. Sorry. Okay. What's a fly's favorite cereal?
Hmm. Fruit Loops.
Fruit Loops. Yes. See, Mala didn't even have the answer to that.
I did.
Testament to her brilliance. So, with that, I will pass it off to the woman, the myth, the absolute legend, Mala Murthy.
Wow, I don't know if I can live up to that intro. Thank you, Amy. It has been a whirlwind couple of weeks. So fun, so exciting. You can keep going. Oh, yes. This is our sort of agenda for today. So I'm going to say a few things and then pass it off to Arie Matsliah, our research scientist and data evangelist at Flywire. And then Albert Lin, who created the Flywire Academy together with Amy. And then Wei-Chung Allen Lee from Harvard will give us an update on the bank. All right. Oh, Sebastian's here. I guess we can let him in. Awesome. Okay. Well, yeah, so I guess I wanted to start off with a bit of you just take a moment to toast the publications of the Flywire Connectome in Nature and the Nature Family Journals on Wednesday, October 2nd. We had a little party at the Society for Neuroscience meeting. So wonderful for those of you who were able to join us there. But now at the town hall, I just thought it might be nice to sort of step back and reflect a bit. And I think we were obviously extremely pleased by the press coverage, as we just said, but we were also pleasantly surprised, I think, by the public interest. The complete map of a brain has really gotten the world very excited in ways I couldn't have anticipated. And I'm sure each of you has now discussed the Fly Connectome with your colleagues in other departments, with your friends, with your family members, and explained your role in the project, which is great. And so I think we should not only celebrate this milestone, but I think we should also reflect on the win for open science. I just want to say a few words about that. Um, so the Flywire Consortium was the last author on the flagship publication, highlighting the importance of the community to building the connectome. And in the grant Sebastian and I wrote way back in 2018 to build Flywire, we proposed not only generating the automated neural reconstructions of FAFB, the EM dataset, but sharing those reconstructions openly with the Drosophila neuroscience community and eventually citizen scientists to both fuel scientific discovery, which I think we have seen we've done immensely well. So, if you go to flywire.ai, you will see the growing list of publications that use the data, including these 12 papers here that were published as part of the Connectome paper package. So we fueled scientific discovery, but we also were able to solicit community help in generating the whole brain connectome. And so once we developed the proofreading platform, Flywire opened to the community in 2020. And the bioRxiv preprints of the connectome were published in 2023, about a year before the official print publications. And so, releasing the reconstructions 3 years ahead of publication could have created issues. But we found instead that when groups converged on neurons in the data set, they often collaborated. And I think one very nice example is on this list from Garner et al. So this was a collaboration between Sung-Soo Kim's lab at UC Santa Barbara and Matthias Wernitz's lab. And those labs came together to collaborate on this very nice publication. We also established an advisory group to deal with conflicts that arose, and I want to thank our advisory group. But we actually never once had to call them in for arbitration. There were occasional conflicts, but that got sorted out on sort of a one-by-one basis. And so I think it's impressive the way our community has come together to build this, um, for this achievement to build this resource to generate all the science. I'm really incredibly proud of the community we've created, and I really hope it's just the beginning for large-scale open science. And so we'll talk more about that today, what's to come. But I think you all should feel, incredibly, happy and proud of this achievement for the community. Okay, that's all I have to say. Let's keep going. Um, Oh, I think Sebastian, if he's here, maybe wanted to say a few words about the press conference we did just following the publications at SFN.
Oh, let's mention that later.
Okay, we'll come back to this. All right, so Flywire updates. So first is we really want to keep track of publications using Flywire brain connectome data. It helps us to understand the impact of the connectome and the resource. So please write to Amy and Marissa if you have publications either in preprint form or journal print form that use the connectome. We want to put it on the website. We want to know about it. The second has to do with new synapses available in Codex. So actually, if you go ahead, Amy, we can come back to this slide in a minute. So Princeton has been working with Zeta AI to build new models for synapse detection. And so the synapses that came from Jan Funke's group that are published as part of the connectome were very good, but they had issues in particular brain areas, or they had issues in particular in some brain areas. And so we developed new ground truth that we trained the new models with together with Zeta. And these synapses are now available in Codex. Arie can tell you more about how they're available. We've done a bit of evaluation on them. So one thing we've done is we've evaluated their performance in these 16 cutout volumes you can see here in the graph. These are the F scores. And blue are the Princeton synapses. You can see that in each of these cutouts, it's performing better overall. Although there are some regions, for example, in the optic lobes and in the protocerebral bridge, where the Buman et al. Synapses actually perform better. And so we're going to make the new synapse detection available to the community. There will be parts of the brain where you absolutely will want to use the new synapses. For example, if you're interested in connections to the photoreceptors, there may be parts of the brain where the Buman et al. Model might be preferred. And so more on that coming soon in Codex. Okay, can you go back, Amy? Back. Oh, great. Okay, so the third thing is that we know that there's data that can continue to be mined within the EM data set that's not represented in Codex yet. And we'd like to get it there, but we're trying to set up some special interest groups who are working on parts of the data like glia or organelles, so that we have a sense for what's being done when the publications are planned, and so that we can make a plan to integrate that information into Codex. So that's item 3. If you are working on data that's not in Codex now, that extends beyond the neurons and synaptic connections present there, please let us know. And then number 4 is that coming up soon, we'll hear from Wei Lee, and he'll give us an update on the BANC, the female brain and nerve core data set. I just wanted to let you know that proofreading, which is being done by Flywire, is around 50% complete, I believe. The VNC is complete, and we're working in the brain. And there's ongoing work on annotations, both cell typing and adding labels that Wei will tell you about. Okay, can you go ahead, Amy? Okay, I think it is now time for me, if you keep going, to pass over to Arie, who's going to tell us more about Codex and new updates.
Hello, everyone. Yeah, so I'll give a few highlights on the updates we made to Codex, as well as update to the recent data challenge on Flywire that concluded last week. Emmy, can you go to the next slide, please? On the Codex updates, first, I wanted to highlight some new data that is now available. First is annotations. Now we're pulling annotations from multiple sources, mostly from papers in the Flywire package that were published 2 weeks ago. So as you remember, initially we were mostly using the annotations from the flagship paper by Schlegel et al. And now we have annotations for the visual system from the visual neuronal pathways of the visual system paper, as well as annotations for the auditory cell types, as well as annotations for double sex and fruitless cell types. This is a work in progress. And we also infer annotations from the community labels that we believe are correct. So all these 5 different sources are now available, and you get a pretty comprehensive set of annotations for all the proofread cells in the Flywire brain. All in all, we have cell types now available for about 99% of the cells in the data set. Then in addition, as Mala mentioned, we have a new synapse table. For now, it is available on Codex to download. We still need to decide how to incorporate it in Codex. We will either use it as a default new synapse table, or we might make some hybrid depending on additional evaluations that we're making. But as of this moment, it is available to download as a one big table, and you can use it for any analysis. Then if you have access to BANC dataset, the BANC is also available in Codex. There's a little drop down on the right top corner. This is the screenshot on the right-hand side. And from there, you can explore the BANC dataset. We are setting up a periodic update mechanism so that this data will be updated maybe on a daily basis because the proofreading and annotations are now happening at a high pace. I want to mention also Alex Spade's contribution to this effort. This was made possible with a lot of help from Alex, especially importing all of these meshes to the BANC space and allowing us to match the different data sets. Next slide. Yeah, we'll also highlight some new features in Codex. One is the visual cell types catalog is now got a lot more details. First, we have a catalog of types for both left and right optic lobes, as well as both optic lobe intrinsic and boundary neurons. Boundaries are visual projection and visual centrifugal neurons. The information on the catalog also contains our subsystem predictions, as well as small snippets of text that explain for every cell type the different properties. Additional feature that is available in Codex is grouping of nodes in the network and pathway tools. You can now group them by cell type, by any other hierarchical annotations. This is for summarizing complex networks and on a more higher level. And lastly, I also wanted to mention a new tool for visualizing taxonomy of annotations. This is the screenshot on the right-hand side. Like many other apps in Codex, you can type in any search query. In this case, I typed in a search query that is for downstream neurons from some optic lobe cell. And then this tool will allow you to visualize kind of the breakdown of all the different neurons in the matching results by annotations. And this is dynamic. You can type in any query and and get this tree of annotations with cell type counts in each rubric. Next slide, please, Amy. Yeah, so since Codex is our primary portal for anyone in the public to explore our data, this is a good way also to measure how we're making impact with this data. And so I've made two sections to this slide. One on the left-hand side is before the publication, before October 2nd. We had about 10,000 users registered globally in Codex. This map was also made before the publication. And since the publication, we got 12,000 new users signed up to Codex, so more than doubling the user base. And we also got bombarded with search queries. At the peak hours, we were doing about six searches per second. Now it's cooling down, which is good because otherwise we would probably run out of funds just to pay for this cloud expenses. Next, Amy, I wanted to give an update on the Flywire Data Challenge. This was the second challenge, and our goal here was to estimate the amount of feedback in the Flywire brain. Feedback analysis is important for understanding certain circuits that are associated with memory or temporal activity like motion detection. And so we have posted this challenge as a combinatorial optimization problem. We estimated with some greedy algorithm that the amount of feedback is at most 31%. And after about 3 months of this challenge being open, 8 teams were able to beat the benchmark. And the best team provided a solution where the amount of feedback is only 15%, which is less than half than the original benchmark that we posted. Now, there is a trivial lower bound of 7% for the amount of feedback. So there's still some gap. And if you are able to improve beyond the best solution, you can, although the challenge is over, but you can still gain a spot on the leaderboard. Feel free to submit improved solutions. Next challenge will be coming soon, and we are thinking to have a challenge on matching connectomes from 2 different data sets. Likely, we will do it for the VNC matching BANC against MANC. MANC is the male nerve cord data set. So details will be coming soon about this new challenge. Next slide, please, Amy. And lastly, we wanted to congratulate the winners of the data challenge: Justin Ellis Joyce, Gert Jan Both, and Srini Turaga from Genelia. They were able to provide the best solution, and we will be hearing from them on October 28th at noon Princeton time. We'll have a short presentation from the winner and the next 3 teams, the runner-ups, who were also very close. So please tune in on October 28th and hear about their solution. And by that time, we should also have the new challenge, and we'll talk a little bit about that as well. So that's it for me.
Oh, and I'll just add, we dropped the RSVP link in chat. So if you want to RSVP to this award lecture, you can just do it on that Google form.
All right. We can go to the next slide, Amy. So I've been working with Amy and Arie with the support of the Science Outreach Office here at Princeton to develop a set of classroom materials that leverage the Flywire dataset and the Codex platform in what we're calling the Flywire Academy. So next slide. So as kind of a very high-level overview, the goal for us was to develop a package of educational materials that could be built around Codex that teachers could use at either the high school or the college level kind of independently of us, so without the need for a neuroscientist to be facilitating. And this would also give the students the opportunity to explore some cutting-edge scientific data. And so we've been working on two sets of resources, one at kind of the AP or IB bio level for high schoolers, and one at the kind of intro to neuro college level. I'll talk a little bit more about those on the next slide. But I also wanted to note that additionally, we want to provide a place where we can kind of host materials that the community has generated through science outreach. So many of us, including us here at Princeton, have used Codex in the past as part of kind of various outreach activities or lessons. And we have a link available online where you can kind of deposit materials and include some information about what level those materials might be appropriate for, and we're going to make them available in a community-facing repository. Next slide, Amy. So the current set of materials we have are basically 3 dry lab lessons available at high school and college levels, so 3 separate sets of lessons. And each of them has been designed to take between 20 to 40 minutes of class time. These are already available online, so you can check them out on the Flywire Academy page on Codex. And they're going to be accompanied by a set of videos, which Amy and I have been working on and will be online later this week, explaining the role of connectomics and the fly in neuroscience. And the purpose of these videos is to kind of explain to students the scientific purpose of the connectome, contextualize that work, and make it possible, we hope, that these materials can be deployed in classrooms in which the teachers might not be that familiar with neuroscience or connectomics. And there's kind of materials we're looking to develop in the future, so additional lessons on more specific topics, as well as programming exercises, especially at the college level. Next slide, Amy. So if you're interested in getting involved with the Academy effort, there's a few things we could use some help with. So first, we have done some testing of these materials, but we're definitely eager to get more feedback on them. So if you're either interested in using the materials, or you know someone who might be interested in using those materials, please let us know and let us know how it goes and if you have any suggestions. And also, if you're interested in working with us to develop more lessons or some, more materials, please get in touch. Here are our emails. All right, and with that, I'll pass it on to Wei-Chung Lee, who will update us on the BANC.
Thanks, everyone. And yeah, please go ahead and advance. And I'd like to start off by thanking everyone in the community, as well as obviously Flywire and folks at Princeton for the tremendous work on helping to proofread the brain and nerve core dataset. Next slide. And so some of you may have seen videos like this. What's being shown is for each edit in the BANC dataset as a function of time, the point at which kind of the edit was made in the dataset. And so, across a little over a year now, well, actually under a year, there's been about 1.5 million edits. You'll see that they kind of start at week 18, which is about when the 611 team and Flywire started working in the dataset. So, and you can kind of see the emergence, right, of a whole nervous system here. So next slide, please. And this is just kind of quantifying that, thanks to Sven, the number of edits per day. And you can see that the dataset entered kind of the chunk graft in the environment back in July 2023, but it was about about December, January, before kind of 611 and the team started proofreading. And you can see the tremendous amount of effort and edits that have been made, kind of peaking across the summer. And then more recently, I think people have been getting to kind of more challenging aspects of the data set. So there's been a little bit of a drop-off as we're also starting to do annotation and looking kind of more deeply at this data set. Next slide, please. And so as Mala mentioned, we're kind of about halfway done if you assume that there's about 150,000 neurons across the central nervous system. The VNC is kind of in the upper left-hand quadrant, which should be pretty much complete. The midbrain is in kind of the right-hand side, kind of the redder colors, as well as the right optic lobe, at least has seen also major progress. And that was kind of reflected also in kind of the edits that you saw previously. So we're a good portion of the way there, and hoping to wrap it up as most efficiently as we can. So over the last few months, we've been looking at doing some initial analysis on kind of the predicted synapses that were generated by Zeta AI. So there's about 230 million synapses that were predicted across the data set, and this is a sampling of just under about 5,000 of them. And this is in green are the cells or the synapses that are true, and the ones that are in cyan are ambiguous, and then there's some that are false in red, and this is plotted as a function of the synapse size, which is a parameter that's output or from the synapse prediction. And so what you can see here is that for the most part, if there's a predicted synapse, they're largely true. There are some false positives, but they're largely correct, but we also know that there are some false negatives, and specifically in certain neurotils like the mushroom body. But what overall this means, though, is that there are synapses that people can use and start kind of analyzing the data set, knowing these kind of caveats. I guess another aspect of this is that we don't see a strong correlation between kind of the size of the synapse right now, at least in this iteration of synapse predictions, and the kind of the accuracy at this point. But we hope to improve that in the coming months. Next slide. And this is work by Alex Bates and Sven Dorkenwald. And so this is Alex, what he's been able to do is to use NBLAST to look at predicted top hits and morphological matches between the BANC and other data sets such as FAFB, HemiBrain, and MAN-C. This is slightly outdated, but the point is the same, which is that largely if you have an NBLAST score of greater than 0.5, they're virtually all correct. And then as you kind of go down in the distribution of NBLAST scores, is if they're kind of greater than 0.3, they're kind of largely good and correct. And then there's some kind of tail kind of with lower NBLAST scores. So the point here is that with high NBLAST scores, we're fairly confident that a lot of those are almost automatically correct. But as you kind of go down, one would want to kind of evaluate on a case-by-case basis, kind of below 0.3, I believe. So if you press the advanced key, and so actually that should be from high scores greater than 0.5, but across the BANC, there's about 30,000 neurons that are provisionally mapped to FAFB cell types, about 11,000 that are mapped to hemibrain cell types, 13,000 that are mapped to MAN-C cell types, as well as another 10,000 or so that have been verified by the Wilson lab. And so ultimately these will be available on a CAVE table. We're kind of processing that now, but what that means is that there are kind of NBLAST results that should facilitate matching, some of which are kind of almost automatically correct, or we believe with high confidence that correct with high NBLAST scores, but at least we'll have kind of a priority list of potential candidates as well to be available soon. Next slide, please. And so what Jasper Phelps has also done is to kind of pull skeletons of neurons from the BANC into both the brain space and the VNC space. I don't have at this point outlines for you yet, but what you can see is that these are color MIPS where you can now or kind of when these are available, be able to search BANC reconstructed neurons against lines using things like Brain Circuits IO or Neuron Bridge. Next slide, please. So turning to annotations, we currently have about 25,000 uniquely annotated cells, some of which with multiple annotations. We would like to increase this number of annotations. And so if people are working with annotations, it would be great to submit your cell annotations by November 15th, which is about a month from now, or exactly a month from now. You can do that through the BANC bot. And if you have questions, feel free to reach out about that.
Hey, Wei, maybe you could just say a bit more about sort of credit for submitting those annotations by November 15th.
Yeah, and so I think the idea here is that if you can get your annotations in, that those annotations that you provided kind of get you, credit, as Mala said, for those cell type annotations. And this can be in the context of publications moving forward. Was there more, I guess, that you were thinking of, Mala, or?
No, that's good. I just
Yeah.
I thought it's yeah, just according to the principles which folks have signed on to, just
That's
reminding
right.
them that contributing both proofreading and annotations will contribute towards the BANC publications to the consortium author list, just as with the Flywire Brain Connectome. And
That's
so,
right.
so submitting them by, if you want credit for those annotations, submit them by November 15th so you can be part of the papers, basically.
Okay, can you move forward? And basically that turns us to, or brings us to kind of the manuscript or manuscripts that are in preparation. And so we do expect to still, or we have a goal of generating, the main BANC paper or preprint by the end of this calendar year. We kind of laid out this goal in the Flywire BANC policy, and we are trying to adhere to this goal, and we think that we can. So be looking forward to that soon. And I think with that, those are the major kind of BANC updates for today.
Great. Well, I think at this point, Sebastian, unless you had something to say, we could open it up for questions. Just sort of overarching, our main goals right now are completion of the BANC connectome and effective dissemination of it to the community. So if you have questions or feedback on that, we'd love to hear it. And then also improving Codex to make it as widely used and useful as possible. And so again, if you have questions or feedback about Codex, we'd love to hear it. Yeah, I mean, I will say that Codex has impressively handled the influx of new users. I've been very impressed with what Arie has created on that front. And so, yeah, I don't know if anyone has experienced any delays, but my experience is that it operates just as it always has, despite the massive influx. So kudos to Arie.
Yes, did you want to?
Go ahead, Nan.
Yeah, I guess since we have a captive audience, I want to ask a naive question. I imagine most people already know the answers, but it's for Wei about you mentioned a couple of circuit level tools for, I guess, and I'm probably going to say this wrong because I actually don't really quite understand the nature of the combined data set. And so I guess the issue that I'm asking is how people are leveraging these tools in order to get merges between the brain and the nerve cord. Or is that already yeah, actually just clue me in, I think is what I need.
Okay, so just to kind of zoom out for a bit. So this data set is a whole central nervous system, so a combined and connected central brain and nerve cord of an adult female Drosophila melanogaster. And what we have now are many proofread neurons with many edits, and there are now tools, or will be soon tools available to help to match these neurons across data sets, meaning matching cells in the BANC with cells in the hemibrain, FAFB, Flywire, or MANC.
You described end blast results, but then later you actually mentioned a couple of other tools that sounded like they in the yeah. So you said neuralcircuits.io and another
Ah,
one.
right. So this is to be able to look up neurons from the BANC against, for example, transgenic fly lines at Neuron Bridge, which is a tool at Genelia, or braincircuits.io. So that will allow you to kind of identify different lines that you can use for subsequent experiments that match up with neurons in the BANC.
The match uses essentially the same uh, architecture as NBLAST to do the
Yes, a very
search.
similar architecture.
Okay.
But you can search against these color MIP images, which are kind of depth-coded images as opposed to skeletons. Does that help? Okay. Yes, I guess I didn't introduce those terribly well, but they're, I think, maybe more familiar with the fly community.
So if while I have the floor, and there, assuming there's a little bit of time, I also was curious about the Flywire Academy, maybe to hear a bit more about how it's being tested. You said you're putting it into practice sort of as you go. I'm curious to know who that's with and what that experience has been.
Yeah, so I guess just the brief answer is some of these lessons are based on materials that I've used at some of the kind of summer programs here at Princeton for undergraduates coming through, particularly in the biophysics summer school. And we've been kind of testing them with kind of various students that we know personally at these different levels, so at the high school and college level. But we haven't yet done a full test in kind of a classroom without us available. So we've also done some runs in kind of classrooms virtually when like I'm around to facilitate, but not a kind of complete test without us there. And so we're kind of working with the Princeton office to set up opportunities for that this coming school year, most likely in the spring, because that's when kind of AP Bio classes get into this kind of material, and we'll kind of evolve from there.
Yeah, and I could also add Gabriella Stern taught a high school class last year, I think, using Codex. I think it was a 2 to 3 day class thing. And one way we're hoping to convince the teachers to actually reach out to us if they're trying this in their classroom is we ask them to email us if they want the answer key. So hopefully if they actually use it, we'll know. Whereas compared with iWire, I know iWire was used in a lot of classrooms, but teachers like never really reached out to us, so we didn't actually know. And I think Arie also has some implemented tracking, so we know at least like how many people have clicked and downloaded the materials. But hopefully more testing in the next year.
Wei, you mentioned that the synapse predictions currently in the bank are really good and people can use them um, already to address circuit questions. Um, but you said that there might be further work on them. Could you say a little bit more about that?
Yeah, so we think that the neurons or the synapses that have been predicted are largely true. But we also know that there are some areas where there are false negatives. And so there are, I think that if you have, strong connections in strong unitary connections, meaning made up of multiple synapses in areas that um, uh, have been sampled well, um, I think that the connectivity that you pull out there should be usable. Um, just kind of note with the caveat that there are areas where things are not predicted well in the sense that there are false negatives.
Oh, sorry, I meant should the community expect that there'll be sort of a new synapse table available within Codex soon or something along those lines?
There will ultimately be like a new synapse table available. The timeline is to be determined.
Good. I think there's another hand up.
Also,
Yeah.
Alex Bates made some comments in chat, so he might want to just say to everybody.
Oh yeah, because I guess Alex, I don't know if you want to chime in, but I guess you had comments about NBLAS.
Oh, the comment I made on the chat was just a, a version of what Wei was saying, which was that in particular it's the Kenyan cell synapses that you can't trust. And that's because the detection is based on Zeta's detection is based on post-synapse morphology, and those post-synapse morphology is less clear in some brain areas, and the one that's very notable is the mushroom body, where they don't look quite so good.
All right, Gert Jan both.
This is Justin with Gary Yan. I actually was going to ask about that because I remember I think the biggest issue, if you look at the confusion matrix of Jan Funke synapse predictions, was a lot of these Kenyan cell synapses were dopaminergic. Do you see that same class difference?
So we haven't yet done the transmitter predictions, and that's something that Diane Adjavon in Jan Funke's lab will look to with the bank soon. She's building a new network to address partly that problem back in FAFB. I think the answer there is partly that Kenyan cell synapses look quite unusual in the brain compared to other ones, and so might better be conceived of as a class of their own. One difference is that they express a particular neuropeptide at the synapse called SMPF, and just in general, the morphology of the synapse looks quite different. So we can probably just learn them as a separate class. If we don't do that with enough training data, I suppose. So in the original network, we didn't throw them in to the training data because we did know that they looked unusual. Yeah. And the result was that they looked dopaminergic, which is false. It's probably because we put lots of training data of dopamine synapses in the mushroom body. So when the network saw mushroom body cytoarchitecture, it just guessed acetylcholine. So the short answer is probably would get the same confusion, but we can learn around that hopefully with the new detection, new classification for the bank.
Cool, thanks.
Congratulations, by the way, Justin and Gert, two of the winners of the Flywire Data Challenge.
Oh, thank you. And thanks to Arie for rounding to the nearest percent, because we get right over that 0.5 bump. It makes it like we won by a little bit more.
Good. All right.
Well, if no one has any questions, I just want to, can we show the slide of the press conference?
Yeah, sure. Let me pull that back up. No, I can't find it. Sorry, hold on.
It's at the end. I put it
Okay,
in sequence, into sequence now.
Gonna say I'm gonna move it. Okay, here we go.
Did you guys get a recording of the press conference?
It was recorded. I don't have the footage yet, though.
So the NIH held a press conference at the SFN meeting, and it was attended by a Flywire consortium, but also John Nye, the director of the Brain Initiative, and Walter Koroshetz, the director of NINDS. And so, one thing about the Fly Connectome is that it really, I think, puts the Brain Initiative front and center. It's a great achievement that the Brain Initiative can brag about. And so the purpose of this press conference really was to advocate for the Brain Initiative. As many of you know, its funding got cut this year. And so I'd like to urge all of you who are US based to reach out to your senator or congresspersons and just tell them about the importance of the Brain Initiative, because it's very important to restore this funding because it's very important research. And so can you show the next slide? And so here we have John shaking hands with Mala, congratulating the Flywire Consortium on our achievements. I think one important message that Mala and I tried to get across was that the fly brain is interesting in itself to study, right? So many of the press tried to portray it as a stepping stone, technological stepping stone to larger brains, which is true. But I think also it's important to let the public know that disease-related genes in humans have homologs in flies, and circuit principles in flies also seem to have homologs in mammalian brains. And not only that, I think the whole approach to fly circuit neuroscience, genetic tools, the connectome, neural activity and behavior, that whole kind of conceptual approach is exactly what the Brain Initiative is trying to bring to mammalian brains. So we were really advocating not only for the connectome, but just for the importance of model organisms like the fly. And so Mala made that point very clearly in the press conference. But it's your chance to, make it clear to your friends and especially to government representatives that this is the kind of research that I think makes all neuroscientists' lives go faster. All right, that's all I have to say.
And this town hall is recorded, so it'll be uploaded on Flywire YouTube later today.
All right, thank you all for coming, and we will see you at our next town hall. Again, send Wei or me or anybody you heard from today questions, feedback if you have it. Thank you all.